It's now my pleasure to introduce our President and Chief Executive Officer, Allan Jones. Allan is going to say a few words to introduce our 15-year retrospective, which is fitting since he's been here from the beginning. Allan joined the Allen Institute for Brain Science in 2003, managing its growth and impact over the first decade. Since then, he's led the launch of the Allen Institute for Cell Science in 2014 and the launch of the Paul G. Allen Frontiers Group in 2016. We are grateful for his steady leadership, guiding us to realize our founder's vision of broad impact in the field of bioscience. Ladies and gentlemen, please welcome Allan Jones.
All right, thanks. I'm going to keep this pretty brief because these guys are the show today. Yeah, so 15 and a half years ago, I left the relative security of a big pharma job to come sign a 3-year contract to work on the Atlas project. Surprisingly, there are 17 people of those intrepid pioneers from those early days that joined me in 2003 or 2004 that are still working at the Institute. I don't know how many of them are here, but I'm going to read the names, and we'll have people stand until I'm done reading the names, and then we'll get some applause. So Nick D. Nick's probably, there he is. Katie Glatfelder. Yeah. Mike Harless, he's right here. We're going in alpha order here. Leonard Kwan. There's Leonard, all right. Edleen. Marty Mortrud. Lydia Ng. Josh Royal. I know Josh is here, okay. Next page. Nadia Shapovalova. She's working in the lab. Okay. Cliff Slaughterbeck. Kim Smith. Nick Stewart. Susan Sunken. Carol Thompson. Paul Wanutka. Wanutka's not here. Brian Youngstrom. There's Brian. All right. Round of applause for everybody. All right, now I'm going to turn it over to Mike, and we're expecting Mike to be on his best behavior. Apparently, he's already been admonished ahead of time. So, anyway, Mike,
thanks.
And Kim and Lydia.
Thank you, Allan. I'd first like to thank our communications team and the showcase kind of organization team for helping us to construct a general kind of audience's version of this talk. Let's see, how do I, let's see, go next here? Okay, great. So it's not really possible to do the full kind of 15-year history in a very limited amount of time. We originally tried by we had 70 slides to begin with, and we had to cut it down, and eventually you've heard about a lot of the more recent stuff, so there'll be really no reason to go into a lot of the great science that you've heard that's been going on in the last few years here. This big product spectrum, we hope to take you through a bunch of this. My distinguished colleagues here will talk to you about a lot of it. A long time ago, in September 16th, 2003, this kind of notice appeared in publicity that Mr. Paul Allen had given this very, very substantial, unprecedented commitment toward what is now known as sort of open science. This idea to profile, to do a large, extremely large kind of systematic kind of profiling of data in the brain, in the mouse brain in particular, to construct what became known as the Allen Brain Atlas. This was not at all a known thing to us. We had no idea really what we were getting into, and it was pretty daunting in many ways. I've got some archival material for folks here. This is a slide here. This, I believe, is like one of the original presentations that actually Allen gave sometime in 2003, in which statements like, we want a path for achieving a high throughput data production capability in Seattle in late 2004. You can see our original kind of original sort of a logo there. We were very closely associated with the Vulcan organization, which helped to serve to provide some infrastructure, some management, some guidance through the leadership of Mr. Allen's staff. We came up with detailed plans and sort of organizational diagrams like this one, which suggested that we knew fully what we were getting into. It's interesting that I'm always kind of amazed about how people, the scale and the requirement for complexity and management, data management publication increases. I must say, in 2003, it was a simpler world. This was an original kind of slide. We showed this slide in several decks. I can remember, I remember Allan giving this presentation, others had given it, but this was just kind of a type of overarching kind of statements. There was a scientific advisory board announcing the commitment. Our original scientific advisory board for the Atlas was really quite notable. The senior advisors, largely, they might have shown up for one meeting, but I think after that they weren't as involved. But this group here was very involved in together with Allan and the team, in terms of kind of directing the Atlas and keeping making sure that the whole thing kept going. Mark Tessiline, now the president of Stanford, David Anderson, who's been actively kind of involved with the Institute and still. Gregor Eikla, whose methods we largely scaled and enhanced here to do the Atlas, and amongst others, Catherine Dulac, whose presence is still in kind of neuroscience with the Institute. We had a website. This was our first web portal here. It basically didn't do anything. It had some news, and if you clicked on these, you got more news. And that was basically the extent of it. It's still a pretty cool picture. This was the original Atlas logo, which I actually see nothing wrong with whatsoever. I think it's perfectly nice. But we changed things around. There's jobs. There were jobs available, in fact, lots of jobs, because we needed people to do this. And I think at the time this was constructed, there were less than 20 people, I think, when this portal was constructed. I think Kim is going to take us over to tell us about some of our old world here.
All right. Well, here's our old friend, Canal View Building, CVB. We ended up being there for 12 years. Interesting fact is that Mike and I kind of came along with the building. There was a biotech in that same building that we both worked at. It folded, and we kept on working at Canal View at the Allen Institute. It went through several construction phases. We started out at just the third floor, then we expanded to the first floor and the third floor, then the second floor, and then the second floor was taken away, and then we went to the Burke, to the Lurk. We finally burst out. Well, actually, there was a fourth location for a little while. Then we finally burst out of it and landed here 3 years ago. This is first, in quotes, structured science. We didn't have that term back then, so this was called the production team. We had one project, one way of producing data. There are some people here that you will recognize. Katie and Nadia and Josh are pictured in here. Susan was the production manager, and this team was responsible for sectioning the tissue, all the reagent prep, and doing the in situ hybridization. What's not pictured is the very small molecular biology group, which consisted of myself and one other tech, and we were responsible for making the probes. And this is a very detailed schematic of the ISH process. Let's see, here we go. So these are all the steps, cascading steps. This process on the TCAN takes 22 hours. And this is the schematic of the actual reaction. There's many reagents. This was our first, this was what we had to get running and to standardize. Tons of reagents. Some were homemade, some are purchased, lot control, automating it on the TCAN. So many factors to consider to creating a quality product. This slide reminds me of something that Allan told us early on as we were driving towards creating a data set to publish for the first time. He gave us the allowance to fail, to just keep on running, make it production worthy, but it is so complicated that you just need to run it and run and run in order to find those pockets of failures and how to improve your data quality. Without that, that allowance to fail, we would have been bogged down in just the minutia of getting everything right without wanting to take the next leap. So it was really liberating, and I've applied that to new projects as they've come along through the years. This is our first collaborator. It's kind of more than a collaborator. Baylor College of Medicine, Greg Reichel. This is where they had this knowledge. They had the knowledge of running a TCAN and doing the high throughput, in situ hybridization on the TCAN and creating the probes. They were very helpful in transferring the knowledge from their lab to our lab, and without this lab, we would not be here where we are today. This is our first microscopy suite. It was in just an office. Paul Wanuka led it up. These microscopes eventually ran 24 hours, 7 days a week when we were in the height of production, over the weekends, and were workhorses. A lot of problems, but they were so essential to our pipeline. And then finally, for this bit for me, the automation. So these are the workhorses. We had, eventually, we grew to 6 TCAN robots. Each run, 192 slides. Depending on how you put your genes, the way that we were running it was 38 genes. We had to get through 20, 30,000 genes for the mouse project, so it's a lot of runs. These were running 5 days a week, 200 slides a day, because 1 run per day, so it's a lot of slides per week. Very essential to have an automated cover slipper when you're dealing with 1,200 slides every single day. All right.
So with 20,000 genes in a mouse genome, at its completion, the atlas will have to, at N of 1, at least have 500,000 images. So we needed some informatics to help integrate the data and to support the search and visualization. Not quite a napkin. I think this is the first known illustration of the informatics pipeline that we can find. And just like Julie's talk from yesterday, the 2 ingredients of this pipeline is image registration to some reference. That's why I was brought in, and quantification of those images that you saw. And that's what Leonard was first brought in to do as well. And we also knew, even at that stage, a 3D reference atlas was key. So in some ways, an atlas captures your current knowledge of what's going on in neuroscience, and it was something that we used to consolidate new knowledge, which is a gene expression, which was then go and improve the atlas. In a lot of ways, the stuff we're doing for cell types now would go and help change the atlases of the future. Hongwei Dong is pictured here. He led the Allan Reference Atlas project, and this is at SFN, in which the publication of the paper version of the Allan Reference Atlas, that's him standing at the Wiley booth for that picture. This is a screenshot of a web application. So now, with more than just news, you can do something. So you can type in a gene, you can look at some images. At that point in time, the team at Vulcan did our web application. Interesting thing to note here is the log out button. So at the very beginning, we had a login system. We asked you credentials, you signed some understand our terms of use, and very quickly, people didn't want that. They just want to come and use it just like Google. So this is how we got to the really open, sort of easy to use access we have today. So you type in a gene, you get to see some pictures. So now we fast forward to 2005, we're generating more genes. And in 2005, we all fit on a single boat for our summer celebration. Who can we see here? We see Marty, we see Cliff, we see Katie, and we see Carol, and Marty again. So then fast forward 2007, This is basically 3 years after we started. We're meant to complete this project, and we sort of completed with a platform publication in Nature, which is a big deal. The picture on the right, it's actually the first figure in which the voxels came into play. Because, well, if you listen to a lot of our presentation, we take these voxels for granted. It wasn't always the case. When we were doing the quantification, the idea was to move those boundaries onto the high-resolution images. We do the quantification there, get our numbers. But because we were making the atlas at the same time, and because the atlas sort of changed every day, it was churning our pipeline like nothing else. So what we did was then we reversed it. We take our gene expression, turn it into some quantification in space. And then whenever someone changed the reference atlas, or we actually wanted to compare some other labels and some other drawings, we could do that. But having voxel allows us to do beginning to do some really cool spatial analysis. So here, up on the top, the correlograms are voxel to voxel correlations. The correlations over 5,000 genes at that point in time. The first one is the correlation gram you get if you just went if you ordered by X, Y, and Z, so from the brain to the back of the brain. The second one, if you ordered a voxel by the reference atlas, so you can see pockets of red because structures are sort of coherent in gene expression. And the last one is like, let the genes speak for themselves. We're going to reorganize the correlation by the genes. So this is genes painting anatomy. And this is really much what we're trying to do in the cell types today as well. And then the graph at the bottom just says that with that, big structures hold because they exist and the gene expression are sort of widespread within that. And then what you see that in the subcortical area, all those areas get mixed up there. What else? Okay. So now the atlas have been out there for 12, 13 years, and it has been instrumental to finding many, many gene markers in comparison. So this is actually from that paper, but even in today's talk and almost each year, you find papers that refer back to our atlas to find certain gene markers. So this is probably one of the sort of legacy of this project. And we also got recognition for this atlas through being in Time magazine, top 10 in medicine 2006, and also recognition in Wired magazine as well. And that's the nice publicity shot at Vulcan with Ed and Paul and Mike and Alan and Mr. Allan and Chen and Carrie, who led the Vulcan team on that web development component. Now we fast forward 2008, and I believe this is the first all-staff photo shoot that we had at that time. And now I'm going to hand that back over to Kim.
Oh, you are going to do one more.
I have one more?
Yeah.
Oh, I do. Sorry. Okay. Built on the success of the adult atlas, our next big project was the developmental mouse atlas. So the intent here was to take 2,000 or so very high-value genes and then survey them in 7 time points. So the trick here is then, not only do we need to make one reference atlas, we need to make 7 of them in order to actually build this pipeline. So this is also wrapped up in the same way and also out on a public website. Now we head to 2009, which we'll hand back to Kim.
And we've grown just a bit on the Canal View steps. So this is a slide for the Allan Human Brain Atlas. So this was a big leap after the mouse atlas. This introduced a host of new problems to solve and challenges, not to mention, the least of which was using whole human brains, working with medical examiners, getting those brains, processing them quickly to preserve the RNA quality, large formats. We had both 6x8 and 2x3 slide introduced at this stage, and a new data modality, microarrays. So a whole new big project, as well as new histological stains. So it was quite an effort and resulted in this great publication. I do remember this is a publication focusing on the first 2 brains, and there was a lot of discussion, how many brains do we need to be able to publish? And we wanted to publish more sooner rather than later, so doing it on 2. We did grow this project to 6 brains, but the first publication was on the first 2. This slide is here just to demonstrate, again, the marker genes, but the transgenic mice that have been a result of our great resource from the Allan Brain Atlas. This is a resource that has continued to be used extensively here, continued to be grown here, as well as external researchers. So a lot of credit to Linda Madison, who's no longer here, but did a ton of work and many years on this project. Connectivity Atlas. We heard a lot about this yesterday through the team talk. It is still going strong. This was their 2014 paper, and it was quite a breakthrough at the time. And finally, we heard about the Common Coordinate Framework earlier today. This was an effort to standardize brain space so that we can map all the data modalities into this space to standardize the analysis, created from 1,600 mice. And it is just so essential for all of our cross-project and intra-project analysis today. Back to you.
Okay, let's see. We've got about 3 minutes to cover 7 years. Yes, the successes of this and the recognition, and it wasn't an obvious thing that, to us anyway, that the Institute would continue. We didn't know on some level, and it was made clear by the founder in his interest and his generosity that of course this was going to continue. And I mean, I think that, you know, we owe a special kind of a tribute to Allan Jones for having shepherded this whole initiative for so many years through this, which really enabled us not only to get this great building, but to get this commitment to pursuing neuroscience in a even bigger way. You know, all this stuff in the past set the stage for a lot of people here who we were very excited to have come at the start of our kind of new plans. This is a picture of with Christoph coming from Caltech, and with Mr. Allan at the announcement in Fremont of the 10-year plan and initiative, and the idea of bringing much more fundamental science into the mix of our operations. And then the establishment of new institutes, the Cell Institute, now immunology, about to be kicked off, I believe, next Wednesday. There's an enormous amount happening, and I'm very pleased and proud to be able to hear with you to actually see how this is kind of realizing it. Here's a great picture. We don't get these kind of pictures all that often, but here's Allan at the White House with President Obama, the great President Obama, Terry Shanowski, other people I don't even know, Francis Collins, certain other people I don't know, but they're I guess. Hennessy. Yeah. Okay. Oh, that's right. Larry Rubin. Yeah, that's a nice picture. That's a. Yeah. We tried to get that decor for the building, but it really didn't. But. So anyway, we're growing, and now it's getting so big that you have to, like, they had to use like a fisheye lens in front of and back of the Fremont building to actually get everybody in the picture. There's something that kind of interesting about this because it makes the whole place look even bigger than it really is. A special call out to, you know, our communications team and all the people who have really been in our program management teams, who have been so instrumental in, you know, a lot of our work, including great SFN, many years at SFN, and many meetings and fun things. Here, by the way, is Stephen Smith holding the world's largest poster session that he managed to sort of give in this last SFN on his thing. You'll hear about his and he's got a poster. And here's a poster from our Southeastern China collaboration with Han Xuanpeng. and they're just sort of displaying their virtual reality stuff, and Hong Kui plays a big role in this too, and others, Amy Bernard talking about product with Tyler, and you can see a lot of activity here. This is the building. It's a big difference in that building Kim showed at the beginning of the talk, and it's almost incomprehensible to us, those of us who lived in the basement of that building, that you might actually be in such a building some years later. Very proud to walk in here every day. This is the unveiling. This is a picture here in much happier times with Mr. Allen cutting the ribbon together with our distinguished internal executives here about inaugurating the building. Probably a lot of you actually were there or saw this. It was a pretty momentous event. 2017, now we're in this building. We're kind of all kinds of creative ways you got to get to make people sort of get the shot work out. But you thought this was the limit, but you know it's not. There's more coming. It gets bigger and bigger. Here's a slide just talking about, we have been playing, just to wrap it up kind of, we've been playing more and more of a role in the community. I think one of the major things the Allen Institute has done has really helped to define data sharing and open science. I mean, I think that's unequivocal. And it's difficult to measure that impact, but it's very much there. And I can tell you that if you've been to SFN and you walk around the floor at SFN, there's a very noticeable and palpable prestige associated with belonging to this resource. This is kind of our more recent involvement with the Brain Initiative Cell Census Network. We've got 2 major awards here through Ed and Hong Kui, plus we've got a center where a lot of people are involved in trying to organize and make this data part of our own. Again, this is what we look like today. You saw from the earliest times, things have changed a lot. There's a lot of functionality. A lot of the product team and the technology teams do a lot to kind of bring all this stuff forward. And here we are, back at where we were in the beginning. That's kind of more or less our wrap up. Of course, it takes this many people now to do it. I can't imagine what it's like it's going up by powers of 2 or something. I can't imagine what the next picture would look like. And then, finally, again, as it's been said not enough times, that this would not really be possible without the vision. That look right there, I know that look. I remember seeing that look for many, many years, and we're going to miss it. I'll miss that look a lot. So thank you very much for your attention and your